whole section, starting with this page, 1017, is about viruses and bacteria and uh-oh, that's not good. Do you need a piece of paper? There's paper in the cabinet. Okay. So this whole section is how do we get infected? What's that look like? So when we talk about viruses, there's a couple of different ways that they can work. So there's a couple of different life cycles that they can have. One is called the lytic cycle. One is called the lysogenic cycle. And if a virus has this cycle, it can be forced into the lytic cycle. Okay, so this figure kind of explains that. So you may remember way back when we talked about figuring out that DNA was the genetic material, that Griffith was a guy who did experiments with mice. He injected them with bacteria. Some of the mice died, some didn't. He was trying to figure out why, what was going on, what was happening. I don't know if that sounds familiar to you or not. It's what my Bio 1 students are currently working through, so I'm very familiar with that. I realize you guys might not be. So if you need a refresher on what that looked like, that was back in the beginning of chapter 10. But essentially, after his experiment, then Hershey and Chase used his research. There were another group of researchers. They figured out DNA was the genetic material. And they did that by using bacteriophages. So bacteriophages are viruses that attack bacteria. So that phage is a term that comes up here, and it's just used to mean something that is infecting. So if it is a phage, it's infecting. So usually it's a virus. And viruses are just infectious particles. They're not even cells because they don't have everything that a cell has. So literally they're like a little container that has DNA or RNA, so some nucleic acid, and it's got protein on the outside. Okay, that container is called a capsid, so it's like the capsule that, bless you, that the virus is contained in, and then it has some sort of mechanism to infect and inject that material into whatever it's infecting, whether that's your cell or a bacterial cell. It has to inject the material into the cell. So when you're looking at this lytic cycle, the phage, whatever it is, attaches to the outside. So this is bacteria. This would be a bacteriophage. It inserts its DNA. The DNA circularizes, which literally means it forms a circle. So instead of being a line, it now is this circle. And in the lytic cycle, the phage is duplicated over and over and over again. It's built and put together. And then, do you remember what lyse means? L-Y-S-E, to break. So the lytic cell gets its name because it lyses the cell. So once all of the phages are ready to go, it breaks the cell that it was injected into, and then there are a bunch of phages that can go infect more cells. So in the lytic cycle, the cell that the virus infects gets killed eventually. In the lysogenic cycle, the cell doesn't have to die. So a phage inserts its DNA, it circles up. The DNA actually inserts itself into the chromosome, so it becomes a part of the bacteria's DNA, which is like creepy. And then the cell duplicates itself. And it duplicates itself over and over and over again. So as mitosis happens, this prophage, which is the DNA that was injected and that it attached itself to the chromosome, that's duplicated a bunch. So then when there's environmental stress, that could be a drought, it could be lack of food, it could be something that's attacking the cell, then that triggers somehow through some mechanism, it's different for every virus, it triggers the prophage genes to become active, and then they turn into the lytic cell. So they start making phages, and then they'll break apart, and then they can spread. So this is one way that a virus can be dormant. You can be infected with something, you can have it affect you, so some of the cells become lytic and spread through your body, and then eventually your immune system fixes it and kind of kills all of the active viruses, but then your cells are still infected with the virus. So you can have a virus be dormant in your cells for many, many years, and then under the right stress conditions, it will pop back up again. So how many of you know someone who has had chickenpox? Anybody? No? Okay, chickenpox isn't as big of a problem because we have a vaccine for it. But do you know anybody who's had shingles?
Yes.
Okay, shingles usually is a disease that older individuals get, but it's a variant of chickenpox. So if you get chickenpox, or if you've had that, Or if you've had the vaccine, potentially, then down the road you could potentially get shingles. Depends on how the vaccine applied or worked in your body. So everybody reacts differently to vaccines too. But shingles, once you have chickenpox, is a virus that lays dormant, and with the right conditions, it comes back up again. So I have a teacher friend who gets shingles once every year, if not every couple of years, just because of normal life stress that she has as a teacher, as a mom, at doing what she does as hobbies and stuff. She gets shingles. It's a regular thing for her because she's had it once. So now there's always the possibility that stress can bring it back up. Okay, so that's how our virus can infect you over and over again. When we're looking at viruses, there are viruses for animals like us, but then there are also viruses for plants. The only section that talks about plants is this little chapter here, so there's not a whole lot of information. Pretty much viruses and plants work the same way they do in animals, though. Okay, so this is an example of just a generic virus of what it would look like. So your virus, again, has genetic information on the inside, and then there's a coating on the outside. The coating helps it infect a new cell. So it sticks to a cell, the coating is dissolved, the cell membrane is attached to the membrane of the cell itself, so the two membranes merge with each other, and then there's a coating or a protein around the DNA or the RNA inside of the virus. Once that dissolves, then now there's RNA or DNA, one of the two, inside the cell. If it's RNA, the RNA also has factors that allow it to start to duplicate itself. So there's an enzyme with the virus that makes the RNA over and over again. From RNA, we get protein. So then you have a bunch of proteins that are made. All of those proteins can help make new viruses. And then those viruses can leave the cell and go infect other cells. Okay, so this is one possible way for a virus to work. So the interesting thing is that when we talk about viruses, some of them are RNA-based and some of them are DNA-based. So some examples of RNA-based ones are the common cold, so the flu that you get a flu shot for, that's usually an RNA-based virus. Measles, polio, and AIDS. AIDS is an RNA-based virus, or it's caused by HIV, which is an RNA-based virus. Some DNA-based viruses include chickenpox, so then therefore shingles, cold sores. My guess is most of you have probably had one of those before, and herpes. Hopefully you guys have never had that before. No, nothing? Okay. Some of you are paying attention, some of you didn't think it was funny. That's okay. So when you're looking at viruses, there's two different ways that really determines, or two different things that determine how quickly you get over a virus. First is how quickly your immune system can fight it off. So if you have a healthy immune system, if you have access to good nutrition, and you're drinking lots of water, that helps your body recover more quickly. The second is whatever tissue is infected, how quickly can it repair itself? So with something like the cold, the tissues that are infected are able to perform mitosis. They can repair themselves fairly quickly. With something like polio attacks nerve cells. Those don't usually repair themselves or duplicate to replace themselves. So if you get polio, you're not going to be able to fix the effects that has caused. Does that make sense? Maybe, sort of, yeah, okay. All right, moving on. Next section. This makes me think of the game Pandemic. So if you're into board games, Pandemic is a game that you play with people. It's not like person against person. You try to beat the board. And the idea is there's a disease that's broken out, and you have to cure the disease. So you have to find a cure, and then you have to actually make sure that the virus or disease doesn't spread. So this makes me think of that because the board is a map, and there are different shapes of diseases that you can fight and try to beat. So there's like 4 different diseases at once. This is a lot more than 4, though. Most of these you may have heard of. So the H1N1 flu is sometimes called, and now I'm going to get it wrong, I think that one's called the swine flu. And West Nile virus is the bird flu. Does that sound right? Avian flu. Yeah, it's called that. So with a lot of viruses, they didn't originate in humans. They originated from another species, and then it mutated enough so that it could infect humans. So that's kind of crazy. So that's where some of the flus get their nicknames from. So the H1N1 is called the swine flu because we think it originated in pigs. The West Nile virus is also called the bird flu or the avian flu because we think it originated with some species of birds or populations of birds. Ebola, Zika fever, those have been in the news more recently than some of the others. AIDS, HIV and AIDS, that was around for several decades before it became something that spread around the world. So it's something that, because of a few different factors that affect the spread of viruses, is now worldwide. Every country has people who are infected with HIV, and then that has developed into AIDS. So you should know, and I'll talk about this again in a minute, but you should know what HIV and AIDS stand for, just because that is something that is so big around the world. It is something that a lot of countries struggle with. So that's one that's not going away anytime soon. That's one I think you should know what it stands for. One of the big questions that this addresses is where deadly viruses come from. So the section here, kind of in three different parts, talks about the three processes that lead to emerging diseases like Zika or Ebola. So the first one's mutation. RNA doesn't have the same proofreading processes that DNA does. So if something is an RNA virus, when mutations happen, they're not corrected as often. So if something mutates, then it develops different traits sometimes. Sometimes mutations are harmful and it just kills it off. But if enough mutations happen, then eventually the organism, that RNA, develops and changes quite a bit. So mutation's one. The second is just contact between different species. So again, a lot of flus, a lot of viruses didn't come originally from humans. But because humans interact with things of so many different varieties, birds and pigs are some, but we raise a lot of other things. We investigate and look at and study a lot of other things. People have a lot of different animals as pets. So if a virus originates in one particular population, it can potentially pass to humans. The third thing is just spreading from isolated populations. So because things like travel, flying internationally, getting different things from different countries is now so much easier than it used to be. It's very easy for a disease to spread from South America to Asia or from Europe to the United States. So that affects the ability of viruses to cause a problem. Make sense? I feel like that's fair. That one's fairly straightforward, if nothing else. Okay, this is the particular section that talks about AIDS. So the AIDS virus makes DNA using RNA. So there's a couple of terms here that are going to be new. Retrovirus is one, and reverse transcriptase is another. So retro usually means like throwback, think like kind of old school. So a retrovirus is a virus that reverses the process of DNA to RNA. So rather than DNA making RNA, a retrovirus uses an enzyme like reverse transcriptase to take RNA and insert that into DNA. So the HIV virus has two different copies of RNA. They're identical strands, and those strands are used to make DNA. So that's what this process shows. So once HIV as a virus infects a cell, the reverse transcriptase makes DNA, and then from that DNA strand it makes the complementary strand. So now you have a strand of DNA that's actually a double helix, like it should be, and then that gets inserted into the DNA of that cell. So now the retrovirus's DNA is inside the host cell, and then that causes problems because those genes are expressed, and it's the cell that's doing it. The virus isn't doing it anymore. Okay. So the reason that HIV is a problem is not the virus itself per se, but the syndrome that comes because of that. So that's why HIV and AIDS are usually said in the same mouthful or in the same sentence, because the virus attacks white blood cells, and then that kills off or cripples white blood cells. So because white blood cells aren't active, then people develop a lot of other symptoms. Your white blood cells are what fight off disease for your body. So if you can't fight off other things like the flu, like chickenpox, like shingles, if your body can't attack those things the way it's supposed to, then you get a bunch of other symptoms, and that's called the syndrome. So it's an autoimmune deficiency syndrome. So your immune system doesn't function correctly anymore. Okay. 1021. This is a really short section. So the only term here is prions. And prions are not even DNA or RNA. It's not a nucleic acid at this point. It's a protein. It's a protein that's folded the wrong way, and so it's infectious. So if you get infected with this, you die, pretty much. We don't have a lot of research on this. This is an area that researchers are still studying. Part of the thing that makes it difficult to study is that it has a long incubation period. So you could be infected with a prion, and it could be in your system for 10 years before you show symptoms. Makes it really hard to track how you got infected, what the source was, etc. The interesting example that they give here is from New Guinea, and people in New Guinea were practicing cannibalism of the brain. So it was ritualistic. So as far as I know, they weren't actually killing people and then eating their brain, but if somebody died, then I think they were eating the brain. It's involved in the ritual. So because of that, if you were eating brain matter, you got infected with prions, and then you got this disease. So don't eat brain, or at least human brain. That's probably step number one. But it's very, very different. I'll say it's very, very different. Honestly, I have no idea what brain tastes like, so I can't say that it doesn't taste like chicken. I don't know. Okay? But it's, again, very different. Not something that we would have to worry about happening here. But again, if you get infected with a prion, then you're pretty much dead. Don't get infected with a prion. Bacteria, prokaryotes. Okay? That's what some of this has been about. So when we're talking about viruses, that's one thing. Bacteria are a problem usually because of the toxins or the chemicals that they produce. So if you get a bacterial infection, it's not that the bacteria themselves are like intentionally attacking you, but the stuff that they secrete, because they're growing in large numbers, that's usually what becomes a problem. So bacteria can take their DNA and transfer it in 3 different ways. We've talked about one already. That's transformation. So that's what happened in Griffith's experiment when he killed the harmful bacteria, but he mixed those with harmless bacteria that were alive. When he injected those into the mice, then the mice died, because the harmless bacteria picked up DNA from the bacteria that was dead, and then it became harmful. Okay, so that's transformation. Transduction is when a bacteria is infected. So that's literally a virus or something that's attacking the bacteria, inserting its DNA into the bacteria. So transformation and transduction are both pretty straightforward. Conjugation, think bacteria sex. Okay, that's what conjugation is. So the bacteria literally grow. They're called sex pili, which are things that help them hold on to each other. They grow a bridge between the two of them, and DNA from one gets transferred to the other. Okay, it's bacteria having sex. And yes, it's been caught on camera. There you go. Okay. As far as these things go, the reason that the conjugation works, the third way to transfer DNA, is that once the DNA is transferred, then recombination occurs. So the chromosome that was in the recipient cell originally exchanges DNA with the donor cell chromosome. So then it gets some new traits. So that idea of recombination or exchanging information is called recombination or crossing over. Crossing over occurs in meiosis. So if you remember from Bio 1 anything about that process, that's just where it happens. We will talk about that later. So we haven't really addressed meiosis at this point. But we talked about mitosis. Okay. Last section. So this is how bacteria can transfer information. So sometimes bacteria transfer an entire chromosome. Sometimes they only transfer a little tiny piece of DNA. So the tiny piece is called a plasmid. Plasmids are little circular pieces of DNA, and they can be integrated into the DNA, so they can become part of the chromosome. So that's what this is showing. Or it can exist as its own separate piece of DNA inside of the cell. So remember way back when we talked about prokaryotes and how many chromosomes they had? And I said they had one. They have one chromosome, but sometimes they have little plasmids too, which is extra DNA. Okay. So in this particular example, there's 2 different types of factors or examples that this section talks about. There's the F factor. F stands for fertility. So the way a bacteria is able to copy a plasmid is largely due to this particular piece of DNA, this F factor. So that means it's able to copy itself and then grow sex pili and grow a mating bridge so that it can perform bacteria sex. If it doesn't have this F factor, conjugation's probably not happening. So some bacteria have this, some bacteria don't. So some bacteria can perform conjugation and some bacteria can't, okay? Our plasmids, this is the medical example or why we care about this as far as medicine is concerned. R stands for resistant. So our plasmids are little pieces of DNA inside bacteria that help them be resistant to antibiotics. So that's one reason why there are so many antibiotic resistant bacteria around now, because we used antibiotics a lot and we killed off everything that wasn't resistant to it. So everything that's left, mostly, is now resistant. So things like penicillin is a really common antibiotic. Tetracycline is another one. Those don't work very well anymore because so many bacteria have these R plasmids, they're resistant to them. Okay? Okay.